Alright you guys, this is video E4.1-9, the last portion of the groundwater pollution lab. We should be looking at our page, procedure part 2 here, and this is where we're going to actually do some testing of the wells in our scenario here. Okay, so as we read through this, and again it's very important to be reading through this stuff, we find that we have a problem here, and we should be following along right here. Mr. Waters wrote us another urgent email, continuing to get calls of concern from residents in the Granite Falls area. So the spill happened over a month ago now, all right, and apparently it's starting to spread. So it says this problem now moves into another phase. You've established the basic geological conditions at the site, so we know about the subsurface under the surface of Earth geology. You know something about the groundwater system. Now you must send your team into the field to collect actual well water samples for analysis. Your analysis will test for the presence of any pollutant from the spill, provide you with the information on the current location, direction, and relative rate, so how fast it's moving for the plume, and enable you to advise the County Conservation Board on which wells are already contaminated with water. So as we look at A, the testing stage here, it says you've now collected the water samples from the 15 known wells in the spill area. Alright, so that's what these guys represent. I'm going to hold it up. That's what these guys right here represent. They are all labeled, well, 1 through 12, and then 13, 14, 15. Make sure you keep them in the appropriate order. I will have a pipette for each well. A pipette for each well. We cannot use one pipette in well 1 and then again in well 2. We'll be contaminating those wells and mixing the solutions. We can't do that. Okay, so I'll have all the caps off already, and there'll be a pipette in every single well. Only take one pipette out at a time to fill your sample, to fill your plate, because you'll be taking a pipette of the solution, say from well 1, and my wells will be set up like this, just as they are on this next page here. And it shows you that you need to put a certain amount of drops into each plate. Okay, so look at the diagram on your sheet and then how these are labeled to make sure you're getting it into the right spot on the spot plates. Okay, so for well 1, I'll take out my pipette and I'll go ahead and I'll put it into well 1 right here. And then well 2, well 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and then 13, 14, 15. And this is the actual spill site. We're not putting anything in there. That's just where the spill actually occurred, right here. Okay. So you'll have a set of these. Notice at the bottom of the previous page, it says that if the well water is polluted, the color will turn a deep magenta when the reagent is added to the sample. I will be adding the reagent. So when you're ready for well testing, flag me down. I'll come around with a little bottle of reagent, and I will be adding that to each of these wells. They may or may not turn a different color. Okay? And it's best to view this from above. So look down on that, everyone look over it, so you'll be able to see some slight differences in colors. It then says the analysis stage, and you have to be looking at this right here. Make sure you read through it, word for word. Do not miss these couple questions at the bottom. Make sure you fill out your data table, indicating the color and the estimate of contaminant level. That was on the first page where it told you it was really contaminated, slightly contaminated, or something like that. Lastly, on this back page, we're looking for some pretty in-depth answers to these questions for some of them. Do not get caught not giving enough detail. Okay? So the keys to this lab: make sure each pipette stays in each well site. Do not mix them, so you're only taking one out at a time. Fill it, put the pipette back, then grab the next one, just so we're sure we're not mixing them and putting them in the wrong wells. Okay? Once you're done with that, and your spot plates are full, flag me down. I'll add the reagent. You'll be able to observe it, fill out the data table, and answer these questions. Okay? If you have any further questions, come see us. Good luck.